Understanding pathophysiology begins with understanding the body's basic building block, which is cell. Cells are composed of various structures or organelles, each with specific function. The organelles are contained in the cytoplasm or cytosol, an aqueous mass that is surrounded by the cell membrane. The largest organelle is nucleus, which controls the cell activity and stored DNA, which carries genetic material and is responsible for the cellular reproduction or cellular division. Cell membrane is a phospholipid bilayer which contain ion channels and receptor molecule. One of the receptor diagram you can see on the screen. One end of this receptor contain the receptor binding site and the other end contain the signaling molecule which transmits signal from uh, receptor binding site to the nucleus. Cell membrane and ion pumps are responsible for controlling the intracellular environment. Organelles responsible to carry out this function is phospholipid bilayer, hydrophilic end on outer and inner aspect, and hydrophobic ends inside the membrane stabilized by the cholesterol. Sodium calcium pump and calcium channel also aids in controlling the intracellular environment of the cell. Receiving signal, cell surface receive signal by cell surface receptor and the second messenger. Protein production and secretion are also the function of cell. The organelles responsible for this function is nucleus, ribosomes and newly produced peptide, rough and smooth endoplasmic reticulum, Golgi apparatus, which aids in protein modification, folding and addition of carbohydrate, secretory vesicles containing protein, glycoprotein product, and proteosome which degrade the defective proteins. Next is intracellular transport, cell movement, and mitosis. Centrioles are responsible for spindle formation during the cell division. Microtubules and filaments are also the components of cytoskeleton. Lysosomes and peroxisome are responsible for degradation and destruction in the cell. Lastly, we have mitochondria, which is responsible for the energy production. Let's look at each of the organelle in detail now. Nucleus. The nuclear membrane contains pores to permit metabolites, RNA and ribosomal subunit in or out. It contains DNA, the nuclear chromatin, which only forms about 20% of the nucleus mass. Next is nucleoli. Ribosomal RNA synthesis and ribosome subunit assembly take place here. Nucleoprotein, example synthetic enzyme for DNA, RNA and regulatory proteins, all made in the cytoplasm and imported into the nucleus. Messenger or transfer or ribosomal RNA enters the route for the cytoplasm. Next is cytosol and cytoskeleton. The nutritious fluid medium that bathes and support the organelles through which cytoskeleton ramifies. Many reactions take place in the cytosol. Cytoskeletons contain microtubules. Organelles such as secretory vesicle or internalized receptor can be transported through the cell via cytoskeleton. Microfilament, which are actin or myosin, these stabilize cell shape and act as contractile protein in the muscles. Then we have intermediate filaments in the cytoskeleton. For example, cytokeratin, desmin, neurofilament proteins, and glial fibrillary acidic proteins. The types differ between tissue and all the structural units. Mitochondria. These are the main ATP energy generating organelles and house of the Krebs cycle and oxidative phosphorylation. They have their own ssDNA, maternally derived, which codes a minority of their proteins. A porous outer membrane and folded inner membrane are also present in the mitochondria. Ribosomes. Nucleus produce ribosomal subunit aggregate into the cytosol and attach to the endoplasmic reticulum or lie loose in cytosol depending on the destination of the protein to be made. Free ribosomes make protein for inside the cell itself. Ribosome translate RNA strand into a correctly assembled amino acid sequence which is peptide molecule. Endoplasmic reticulum ER, also known as ER, is an irregular maze of membrane-bound tubules, sacules, and cisterns which ramifies through the cell. There are two types of endoplasmic reticulum in the cell. Number one is rough ER, which is studded with ribosomes. Proteins made by the rough ER pass into the rough ER cistern and undergo secondary folding and early glycosylation before being incorporated into the membrane for export from the cell. Receptor molecules on the cell or components such as liposomes within the cell. The other form of ER is smooth ER. This is further addition of carbohydrate moieties to protein folding to achieve the tertiary structure. Secretory vesicles. These membrane-bound packets are moved via cytoskeleton to fuse with the cell membrane to expel their content outside. Lysosomes. These are intracellular membrane-bound vesicles containing destructive chemicals and enzymes which fuse with phagosomes to release their content into phagolysosomes and destroy pathogen. Lysosome also degrade worn out cells as we said before by the process known as autophagy. Peroxisomes, these small membrane bound granules contain oxidative enzymes which make hydrogen peroxide plus its regulator catalase. Proteosomes. Proteosomes identify defective protein and degrade into their component peptide and amino acid for reuse by the cell. Portions of broken down protein are bound by MHC class 1 molecule and displayed on the cell surface to the TC cell or T helper cell. Centrosomes, this contain the two linked centrioles from which microtubules radiate into the cell. The centrioles duplicate and migrate to the opposite end of the cell during cell division, separating the duplicated chromosome. Membranes. Membranes are phospholipid barriers surrounding the cell itself and the certain organelles. They isolate portions of cell and permit several often incompatible metabolic processes to take place simultaneously. Cell membrane, phospholipid bilayer interact with extracellular world by assorted surface molecule. The center is lipophilic and surfaces are the hydrophilic with cholesterol as a stabilizing spacer between them. The raft theory suggests that intermembrane structures can float and be cross-linked around the perimeter of the cell. Membrane proteins. Proteins that project through membrane outside the cell usually have attached carbohydrates. Glycolipids are carbohydrate attached to the lipid membrane and are important in cell recognition, cell to cell bonds and absorbing molecules. Some tissue have a protective glycocalyx. Transport through the cell membrane. The main mechanisms are as follow. Passive diffusion needs only a concentration gradient, for example, lipids and lipid soluble agents like ethanol. Facilitated diffusion, the binding of molecule trigger a conformational change which moves the molecule across the membrane. Active transport against a concentration gradient to maintain ion concentration within cell, for example, sodium and potassium ATPase complex. Bulk transport, endocytosis, transcytosis, exocytosis. Endocytosis include receptor-mediated endocytosis, ligands and viral particles, and phagocytosis, engulfing of particles. Pinocytosis is the small sampling of small quantities of extracellular fluid which is not receptor mediated. Transmission of messages across the cell membrane by lipid-soluble agents which diffuse directly across the cell membrane, receptor binding and activation of secondary messengers applies to protein messenger molecule which bind to a specific cell surface receptor, resulting in active transport of the molecules through the cell membrane or triggering of the intracellular cascade reaction. Last is neurotransmitter. These are the chemical messengers for neurons or myocytes that cause an electrical response in target by the receptor mediated opening of an ion channel. This brings us to the unit 1 from pathology at glance. Thank you for watching. If you like my video, don't forget to subscribe for more.